Perfect. Neeraj, I want to turn to you based on what you've heard so far from Meinhard and Lena. We know that most of your experience has been a little bit later stage. So you've been exposed to early decisions, I'm sure, on multiple occasions, and had to make adjustments on the back end. So share a little bit about that. What's been your experience with how these early decisions impact later stage development?
Yeah, thank you. Yeah, so I think it's understated many times in the drug development how big an impact it has, these early decisions, like the way Lena was pointing out. I think if you were to do, for example, just as an example, if you were to do, even during the clone selection process, if you have a really nice DOE, to Meinhard was saying, and you run through an extensive, let's say, a product quality analysis, you can understand a lot of things about the process and how robust it can be. So those things can really help you at the later stage. So what I am dealing with, and I'll talk to specific to my experience, development experience, our molecule, it was developed, I would say, good 15 years ago, and we in-licensed it. But there are a number of things we're doing. We are in phase 3 right now, and we see that the productivity is not optimal. And also the downstream process, given the complexity of the molecule, downstream process could be further optimized. And also there are some of the, I would say, CQAs, the way we understand it, the amount of control strategy could need to be slightly different. So we kind of see issues, some of the things that were made decisions way back like 10 years ago or so are really affecting us in a big way. So what we're doing here is that since the regulatory scrutiny, as you know very well, I think all of the audience, I would imagine they know, as you get closer to phase 3 and beyond, the regulatory scrutiny is going to be very high. So my recommendation for any company that trying to do like later stage changes would be, getting an alignment with the regulatory bodies, whether it is FDA, EMA, so that you're in a good shape. So as far as what we are doing currently in terms of how that is affecting, we are exactly doing that, going to the regulators, getting the alignment, and developing approaches where we can bridge pre and post change. So that's what we're doing. So essentially, if I were to be working with like early stage people like Lena, for example, I would go straight to the, even kind of a little bit downstream compared to host cell selection. Even during the clone selection process, I would have this extensive analysis where I understand the failure modes, and so that I can set up pretty robust process, whether it is cell culture or understanding how my impurities need to be purified so that I can set up my downstream process and going into even the control strategy. So that's, I would say, kind of in general, but happy to if anyone else has questions. But, yeah.
Yeah, we're getting some good audience questions as we speak, and some of them are right in line with some of the questions that I had for the three of you. So let's take a couple of those. One of them from Rakesh is, what are the attributes that are monitored during the cell line development process? Lena, I'm going to pick on you to address that one right now.
Yeah, I can definitely do so. First of all, it really depends on the final product. So first of all, you need to define when you're having a product which might be critical parameters you analyze. I think more general ones would be definitely aggregation, because a lot of them are going into aggregation, and I would definitely already screen that on pool level. So aggregation, charged variants, as well as also glycosylation in that part. So I would definitely recommend to already have a look to glycosylation, so N-glycosylation, especially when being in the early part, so already on the pool level. That is really important. When going into ADCs, that was also already mentioned, I think, by Neeraj. or by was it Meinhardt? That you have a look directly to the conjugation efficiency, because what we also see that especially the post-translational modifications can definitely influence conjugation efficiency. So I would already recommend on the pool level to have a look to the conjugation for ADCs. That is definitely, I would say, the main part. But you can already also go to, depending on from the product, for example, to binding assays, so bioassays, binding assays at the pool level. I would say be as extensive as possible already on that pool level for such a kind of complex project to find a pool which is already good fitting on pool level. Then you can go to the single cell level, single cell cloning level, and also on clone level. I would recommend to use already that panel of assays I already mentioned here. So this is because you will see differences also in clone level. I think with novel technologies in the in this biotech field, also with transposase systems integrated into single cell cloning, you will get a little bit more homogeneous pools and clones in generally, but there will be differences. And that I would definitely include also on the first clone screening level. So screen a lot of clones here. So not only screening like 24 clones, but perhaps also put that a little bit broader to really find a really good fitting clone, which definitely fits best to your requirements. And that is what I definitely would suggest to do on that level here.